Hi everybody, I'm Professor Steve Forsyth, and today I'm going to do another video which I've called Advanced Whole Genome MLST Analysis. And as an example, we're going to be looking at strains which have been isolated in the USA 2021-2022, and in particular we're going to be looking at the meningitic pathovar clonal complex 4. Why do this presentation? Well, similar to the last one, currently, you can see the date 25th of March 2022, there's still a lot of social media alerts on Cronobacter and infant formula. A number of questions are going on, and the idea of this presentation is to show you advanced techniques in MLST analysis, but also to put a context as to where one can actually isolate Cronobacter. In order to maybe understand more about PubMLST and looking at Cronobacter sakazakii and taxonomy, then I would advise you to go to YouTube and see the one which I did there a few weeks ago, which will give you that background, and I won't be overlapping very much with that one in this slide presentation. So the structure, we're going to look at the introduction to the PubMLST database, just show you some different angles that you can approach it from. Then I'm going to show you how to download whole genome isolates from the database. We're going to do a working example of the meningitic CC4, look at some conclusions, references, and who am I. Now, with regard to Cronobacter, one hears quite a lot of it with regard to infant infections, but in actual fact the majority of cases are in adults: bacteremia, septicemia, urosepsis, wound infections. There's been one outbreak attributed to food, whereas in infants is where there's a more severe response with regard to infections, and they're primarily in infants who are pre-weaning, those who are less than 6 months in age, especially those that are low birth weight, and the clinical symptoms can be necrotizing enterocolitis, septicemia, and unfortunately even meningitis. Now, where can we find Cronobacter? Well, this slide summarizes some of the diverse places where Cronobacter can be found. It can be found in water, and we're going to come back to that source on the next slide. We can find it in animals such as mice and chickens. We find it in insects. We find it a lot in vegetable matter, salads, herbs and spices. And one which I used to like doing class practicals with was flavored teas, because the tea bag is nicely about 2.5 grams. But because plant material is used as a major ingredient in many foods, you will also find it in packet soups and cake mixes, as an example. But those are what an infant eats. With regard to the infant situation, then the sources can be powdered infant formula, and here we're dealing with infant formula manufactured for infants less than 6 months in age. It has been found in the water, bottled water, being used to reconstitute infant formula. It's been found in the nasogastric tube, because bear in mind these infants are often premature, so they don't have the suckling reaction, so they have to be fed by nasogastric tube. And infants not fed infant formula have been found to have Cronobacter in the nasogastric tube. It's found in breast abscesses, found in breast bumps, and it's found in follow-on foods, weaning foods, follow-on formula, which is where the target age is over 6 months. So why use MLST? Well, if you want to see the comparison with serotyping and pulse field, then please see the previous video. But to summarize, PubMLST started with regard to using 7 housekeeping genes, and at present we have over 700, almost 800 sequence types across the genus, which is composed of 7 species. Cronobacter sakazakii itself has got over 300 sequence types, and it's a very expansive database, so most weeks I'm adding several more strains to the database, and the analysis can be expanded as well. It's actually undergone relatively few tweaks over about 10 years, which surprised even me. Major tweak is that there's a second suggested PCR primer sets for those people who are still doing 7 loci PCR. But the thing is that these days it's totally compatible with whole genome sequencing. And that's most of the isolates that I update into the database these days. And because of that, we have a MLST variation, which is what we're going to focus on today, which is looking at core genome MLST, which is almost 2,000 loci. And you can choose for yourself which loci you want to compare strains with. But we're going to focus on this particular opportunity, which is a default setting in the database. It is centralized database. It's curated. I'm the principal curator for the Cronobacter isolates in the database, but it is open access. Anybody can look at the strains, anybody can submit strains, albeit I will then curate them. So the reason why MLST has been so useful is not only for genotyping, but by looking at the sequence type and then looking at the clinical symptoms. Several years ago, we realized that there were actually definable pathobars. So the ones of particular interest were the clonal complex 4, which means sequence type 4 is the main one, found most often, but you get single and double loci variants. They'll be in, that's why we call it a clonal complex. And we found quite often the meningitic cases were due to this particular sequence type, clonal complex type. Those babies, unfortunately, with necrotizing enterocolitis, we found a predominance with sequence type 12. And with regard to adult infections, it's primarily Cronobacter melanoticus, different species, and it's the clonal complex 7. So Susan, Joseph and I published that over 10 years ago. And here, for those who aren't familiar, these are the 7 species of the Cronobacter genus. When we go to the PubMLST database, and the link is here, if you can't find it that easily, we have a map. The majority of isolates have come from China, so it's like a density map here. If you were to submit your strain, then just click on here, and that can be a whole genome sequence, or it can be 7 loci sequences, whatever, still comes to me and I curate them. If you want to look at the primers, they're here, and bear in mind that there are 2 suggested primer sets now. And if you want to look at video guides like this one and related ones, then just click here. The time of making this video, there were over 3,000 isolates in the database and over 1,000 genomes. We're initially just going to click here with regard to the isolate collection. And when we do that, this page opens up with a lot more data. And what we want to do is go over to the search on the right hand side, click on the plus sign, so you get these 2 options, and you want to click on searchable database, search database. And this window opens up here, this upper part of my slide. And I just simply, for this example, clicked on search, and this is a screen grab of the beginning of the page. And one thing I want to emphasize is that the blue number down here, that's the ID number, not the strain same as the isolate number. The ID number is the order in which they were entered into the database over the years. And if you click on that blue number, then what happens, so this is clicking on the blue number 5, it opens up with further information. So it tells you that this strain 5, isolate 658, it's also known as ATCC BAA 894, tells you the predicted serotype, etc. And also the source here is non-infant formula, and there's comments over here just explaining that this is one of those misunderstandings in literature, that people think that this strain was from powdered infant formula. This is the one from Tennessee, published by Hemorite, but in actual fact it was non-infant formula that was used. So there's information and there's like links to publications that that strain has been referred to, and down here it tells you about the contigs and about the genome details, because this happened to be one of the ones that's been genome sequenced. If we go back to that home page and instead we go to the genome collection, then it's only going to list the strains for which we have whole genome sequences. So that's going to cut out 2,000 of them. But those are the ones for this video I want to concentrate on. So if we click on that, same process, we get a list of strains, and again you can click on the blue ID to get more information, and you can see on this screen grab how I have the laboratory isolate name that's been submitters have told me, and sometimes those strains also have an ATCC number, etc., and that's also entered into the alias. So I try to make it as accessible as possible. So you've got your country, serotype, etc. listed here. It's very important to know the difference between ID and isolates. So ID is the order in which they went into my database, and the isolate will be like a laboratory number by the submitters. It's very important as we progress in this video. So you want to scroll down, and when you get to the bottom, you can see some analysis tools, and there's always different ways of getting here. This is simply a way which I find works for me very quickly. So we can click here for data set, and if we do that, this window opens up and we can download all the information that there is on whole genome sequence strains of Cronobacter and related species. This is all open access. There's no charge, there's no registration or anything. It's just there. So the default would be list all. This is where you want to be smart and just think, what information do I want? And I would recommend selecting ID, isolate, alias, country, location, year, month, and comments. Get those. And for now, just click on MLST for the sake of my demonstration today. So when you click on that, you get an Excel spreadsheet. So here's ordinary Excel. Just go to the sort and filter and reorder according to clonal complex. This is the trick that we're doing here. When you do that, so this is just part of the Excel spreadsheet. I've done it according to clonal complex, and you can see all these clonal complex 4 strains. They're from a range of countries. Okay, Denmark, Czech, Belgium, Austria, USA, Slovakia, Israel, New Zealand, say China, Switzerland. Range of sources: ready to eat food, food ingredients, clinical environment, etc. Range of years, and then we've got comments. And I really would recommend that you at times spend time looking at the comments. The screen grab doesn't do it justice because source might say ready to eat food, but when you click on comments, it tells you which ready to eat food, and it may tell you the SNP cluster number. It may tell you the AMR profile as well. It can all be there in the comments. And we're looking here at clonal complex 4, and you can see there's a range of sequence types. Most are sequence type 4, but there are other sequence types which are in the clonal complex because they just vary by 1 or 2 nucleotides, and that means 1 or 2 loci will change as well. It's a very minor opportunity. So we've got that Excel spreadsheet. We've ordered it according to the clonal complex, and obviously this screen grab doesn't do justice to all the clonal complex 4s that you'll find in the Excel spreadsheet. And then what we'll do, we'll go back to the home page, click on Genome Comparator. That opens up this page. You can then go to that Excel spreadsheet and say, right, I'm going to just copy and paste the clonal complex 4 strains. So clear, and then copy and paste into this window. Then for the output, and this is again somewhere that you'll learn what's best for you. I would suggest something like isolate, country, region, source, and year. And if you think ahead, what we're going to be making is a phylogenetic tree. and you want to think about what information do I want on the end of those branches that will be useful and informative, and that will vary according to your actual purpose. So this is what I'm suggesting for this demo. Then what we're going to do, we're going to do COG-MLST, which is 1836 loci, and we just got to press submit. Now, if you want to take this to a further level, you can, rather than doing the default strain, you can actually put in there what strain you want all these other strains to be compared with. So you can choose your own reference strain, but that's outside the scope of this video. So let's pretend we put in 100, 200 strains in here. We press submit. After 1 minute or less than 1 minute, in fact this example took about 18 seconds, you'll get a screen like this. And for our purposes today, we'll ignore most of the output. You'll see here we got a bit of a nice tree coming out. It's too small, you can't do too much with that image. What I find much more useful is to go to this distance matrix, save that file, and then what I use software called Splittree, and the link is here telling you about Splittree from University Cronobacter, and you can click here to download the software for yourself. So this is the software that we're going to use. So that file you then open up in Splittree, and you can see the interesting way that clonal complex 4 breaks up into various other subclusters, and you can see the end of the branches is where that information is. So that's why I was saying about be clever in what you want, what information you want at the end of your branches. Now, there's about 150 strains of CC4 in this diagram. We need to zoom in a little bit more. So in this upper bit, when we zoom in, we're going to zoom in a little bit more, and we see this. We got 2 strains here. They're both in USA, one's clinical and one's infant formula, and they are indistinguishable. Over here we've got 2 other strains, one's clinical, one's environmental. Again, they're USA, and again they're almost indistinguishable. When we go to look up the background to those strains, we find that these 2 strains are from Minnesota, and those are the information. You can see one's clinical and one's infant formula. So that's quite informative. We know the date, the month when that one was isolated. We don't have a date for the clinical one in the database, but we can also see these 2 over here. One's clinical and one's breast pump, and that one was Kansas, and that one seemed to be collected in March 2022. So what we've found are 2 pairs of strains, one from infant formula that is indistinguishable to a strain that's a clinical isolate, and a strain that's again a clinical isolate, and one that's from breast pump. Now, just to highlight something, there's only so many terms which can be used. So in the database it says environmental. That doesn't mean soil and water. It can mean manufacturing environment, or in this case environmental means it's from a breast pump, because that's not clinical in itself. It is, so there's that sort of ambiguity, so be careful to look at the comments to get more information. Now these are CC4, these are Cronobacter sakazakii sequence type 4 strains, and that's a very important pathovar. It's often found in infant formula manufacturing sites, countries such as Australia, China, Germany. If you want to delve more, I recommend you use the strain which is ID 446. It's also the original laboratory designation was SP291. It's isolated and sequenced 2013 because it was a very persistent thermotolerant strain in manufacturing site, and later on it's found to be ST4. Now, just to show that it doesn't happen only in CC4, I did a quick example. You're looking at clone complex 8, and again we get, when we do the COG analysis, so CC clone complexes, they're only on 7 loci. When we do over 1,800, we get greater differentiation and clustering. And again, if we zoom in here, we find we've got 2 strains. One's from what was in the database was put down as open formula, so presumably that might mean an open tin of infant formula, but there isn't the detailed information there. Isolated, again, Minnesota, October 2021, and we have a very, very closely related strain, which is from bottled water. So that is really interesting. We got 2 strains, almost indistinguishable, one from open formula and one from bottled water, both in the same state, both isolated, we believe, from the same month, which was last October 2021. So you can explore, okay, I've just done some simple examples, choosing some clonal complexes and then just zoomed in on the clustering. You can do this for yourself. What you can do if you find the results, well, at the end of the day, to me, we're dealing with neonatal health in particular. This is a very serious matter if you do find pairs of strains. The ones I've shown you are ones which probably the authorities are already aware of because they were sequenced by the FDA and then they get uploaded. But I would recommend that if in doubt, then contact the officials. So you've got CDC over here and you've got FDA over here. In actual fact, these are live links here. You can click on these to go to these websites. So I think it'd be appropriate if you find pairs of strains which may not have been linked in the past, then let the authorities know about it. Now, MLST isn't the only means of genotyping Cronobacter. There is SNP analysis, there's CRISPR-Cas array profiling as well, and a very interesting one is looking at the capsule composition, the K antigen 2 colonic acid type 2, and with cellulose. That seems to be a capsule profile most associated with meningitic pathovar. So to end this, what we're going to say, well, Cronobacter clearly is found in a variety of sources. The PubMLST offers an open access resource to looking at whole genome sequence strains. And we looked at using COG-MLST, which is a almost 2,000 loci, and what we're able to show quite easily using just free software, strains which are indistinguishable or nearly indistinguishable from clinical formula sources and so-called environmental sources. And just a point at the bottom, indistinguishable does not mean the same. So that's why I talk about the other genotyping methods as well. So to finish off, so to speak, who am I? Well, you read this for yourself. I'm so-called retired, and obviously not very. I've been an advisor to the WHO 5 times on the safe feeding of infants, and various guest lectures here and there. Over 150 peer-reviewed papers, and over 100 of those are on Cronobacter, and the curator of that PubMLST database for Cronobacter with a worldwide network of collaborators. If you want any particular videos or you want a point clarified, please email me here, and most of my papers are open access, so you can just click here and go to my papers. The papers in particular supporting this video are these 6, and again quite a few of them have links so you can access them for yourself. So thanks very much. I hope you enjoyed this, and take care.